Hello, everyone. Welcome to today's broadcast, Non-Destructive Moisture Determination for Lyophilization Process Development and Product Quality Testing. I'm Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be the moderator for today's event. We're pleased to bring this webcast presented by Pharmaceutical Technology Europe and sponsored by Lighthouse Instruments. I'd like to share a statement from our sponsor. Lighthouse Instruments is the leading global provider and manufacturer of Headspace analysis platforms and measurement services. Lighthouse offers products and services for Headspace container closure integrity testing, packaging development studies, oxygen monitoring, moisture and water activity determination, and microbial contamination inspection of media vials. Rapid, non-destructive laser-based Headspace analysis is a powerful analytical tool for generating science-based statistical process and product data at all stages of the product life cycle, from development to manufacturing and quality control. I have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, just click on the question mark help widget in the dock at the bottom of your presentation window. Now I'd like to introduce today's speaker. Dr. Derek Duncan began his career at the Dutch Institute for Atomic and Molecular Physics in Amsterdam. He moved into industry holding product and application development positions. At Lighthouse Instruments since 2003, Dr. Duncan is responsible for developing applications for process monitoring and finished product inspection. So thank you for joining us today, Dr. Duncan. I'm going to turn the presentation over to you.
Thank you, Rita, and thanks to everybody who is participating in today's webinar. I can see that we have attendees from all around the world, so certainly appreciate you taking the time for this webinar. Before I get into the webinar content, I wanted to go over a short history lesson in moisture determination methods used in the pharmaceutical industry. So 30 years ago, for all of us who can remember that, the standard method for moisture determination, also for freeze-dried vaccines, was gravimetric or loss on drying method. And in the early 90s, titration methods started to be used. So there were people who saw some advantages in using titration over loss on drying. And so the titration method started to be used in the early 90s, but the absolute moisture content as measured by both methods was different. And so people at first were a little bit confused by that, but they started with some correlation studies. And so here is an example of that. This comes from a publication in 1991. And as an example here, if you look at the black triangular data point, when you measured that vaccine, the rubella vaccine, with the gravimetric method, you got 0.5% moisture. But if you used the Karl Fischer titration method, you got 1% water. So there was a little bit of confusion about that, but generally people figured out that it is because these two methods are measuring different types of water. And you can see that they do correlate with each other. The correlation is not fantastic, but they do correlate. And so that convinced people to step over from the gravimetric methods to titration methods. And I imagine that the largest percentage of moisture measurements done today in the pharmaceutical industry is done with titration. So that was about 30 years ago, and now titration has kind of been the standard method that has been used for the past 30 years. Now, these standard moisture determination methods that are used in the pharma industry do have some disadvantages, as people have found out. The risk of moisture contamination can be high. You need to handle these samples, so there is a risk of contaminating them. You have to be careful of the environment that you work in. These methods are therefore somewhat dependent on operator skill. The methods are time and resource intensive. I think QC labs are always backed up with their titration schedules at large facilities producing dry product. And lastly, the methods are destructive, meaning that your sample is lost when you analyze it. And for high value samples, such as some freeze-dried samples, this is not advantageous. So because of these disadvantages, it's very difficult, and some people might say impossible, to analyze statistical sample sets of finished product. And if it's very challenging to do that, that means that insight into the drying process is a little bit limited if you're relying on the analysis of finished product. And also the accuracy of moisture stability specifications is also limited due to the destructive nature of the measurement and also assumptions about identical stability samples. So first, 30 years ago, loss on drying was the go-to moisture technique that changed to Karl Fischer titration. There have been introduction of new techniques such as NIR. This has not fully caught on for various reasons. And so the question is, after 30 years of using titration, is there a better way to do moisture determination of dry product, and in particular freeze-dried product? So that is a question that this webinar agenda hopefully will address, and I'll start with an introduction of a possible technique that could be useful, and I hope these case studies do show that it is useful, and that's Headspace moisture analysis. We'll compare this technique to the standard moisture methods. I'll also briefly describe the relation to water activity and give a few application examples, and then we'll spend most of the presentation on real industry case studies, which will have examples from different points of the product life cycle, from formulation development to process optimization and scale up, and then manufacturing and QC. So Headspace moisture. If you have a freeze-dried vial, the total water content is not only the water content that's in the actual lyo cake, but it also includes the water in the headspace, or the water vapor, the free water in the sealed vial. And if you look at the different techniques to determine moisture, they all measure slightly different things. So Karl Fischer will measure the total moisture content in the vial. If you look at loss on drying and NIR as well, that will measure cake moisture, the water that's in the cake, or some portion of the cake. And lastly, laser-based headspace analysis will measure the headspace moisture, or the water vapor pressure that's in the sealed container. And this water vapor pressure is a measure of the free water that's in the sample. And actually, all of these measurements do give an indication of how well that sample is dried, and you would also expect that these measurements should correlate with one another. So here's an example of a correlation, and here a set of samples were analyzed over a range, and these samples had a range of moisture values. First, the samples were measured with headspace analysis, and a water vapor pressure was determined. That's plotted on the x-axis. Because the measurement is non-destructive, these same samples could then be titrated, and you see the Karl Fischer values on the y-axis, and you see quite a good correlation here. In this particular case, it was a sucrose formulation, and you see an R squared of about 0.95. If you'd like to see more examples of this and details, there's a reference there for you where this data was published. So these two measurements, let's say the headspace water vapor measurement and titration measurements as done by Karl Fischer, do show some good correlation with each other. If you're not familiar with laser-based headspace analysis, it's an analytical technique that was introduced by Lighthouse a little more than 20 years ago with the help of some FDA funding. And what you do is you simply shine laser light from a laser diode through the headspace of a container, of a sealed container, and that allows you to measure specific gases in the headspace. And depending on what the frequency or wavelength of the laser light is, you will detect or measure and quantify different types of gases. This measurement is non-destructive and it's very rapid. It takes on the order of a second to make a measurement. And this flexibility and ease of use of this technique makes it ideal for rapid non-destructive analysis of finished sterile product. And Rita mentioned a few applications in the introduction. Container closure testing can be done with this technique, headspace oxygen monitoring. But for the moisture application, what it does is it determines the water vapor levels in the container. And we'll go into this in a little bit more detail, but it's also described in an upcoming, in a new USP chapter, USP 922, about water activity. So it can be used to determine the water activity of a material. Here are just some pictures of some headspace platforms, just to give you an idea of what they look like. On the left-hand side, you see a benchtop headspace analyzer. These are implemented in laboratories or at line in production. It's a very simple instrument, easy to use. On the right-hand side, you see an automated headspace inspection machine. These can be implemented in line in production and can do full batch inspections if desired. So let's go over a few examples for applying the headspace water vapor measurement as it relates to lyo products. So in this particular example, we're going to show that water vapor levels can correlate to the appearance of a lyo cake. In the upper right-hand side, you see at the moment a graph. This graph is plotting the headspace moisture values of 20 samples. And you can see that the first 10 samples have relatively consistent low values of headspace moisture. And if you look at those 10 samples, pictures of these 10 samples, you can see that the cake appearance looks okay. This particular formulation was a very difficult formulation to freeze dry. There was no buffer in it. It was a hard process to control. But you see that these 10 vials, the cake appearance looks fairly good. If you go to the next 10 samples, and here you start to see a sharp rise in the headspace moisture values, you can see that all of a sudden you start to see bad cake appearance, and the bad appearance becomes increasingly bad as the water vapors increase, so as the water vapor levels increase. So we've had customers apply headspace water vapor measurements to pick out lyo cakes that might be defective in a sample set simply by flagging the high water vapor vials and pulling them out and inspecting them a little bit more carefully. Another example for using water vapor measurements is it can enable robust and efficient lyo cycle optimization. So if you're convinced that the headspace water vapor tells you something about the product dryness, then you can use it, for example, to perform 100% moisture analysis of a pilot batch. And that kind of data is plotted here. So we had a client that produced a pilot batch of roughly 1,300 freeze-dried vials. These vials were in trays in the freeze dryer. So here you see a plot of the headspace moisture values per tray. And what you see is that the batch is not really homogeneous in the headspace moisture values. And in fact, the values are location dependent. So in tray 2, you see that samples dried a little bit better on average than in tray 4. And so when the client saw this, they wanted to try and improve the lyo cycle or optimize it. And so they changed some parameters in the cycle and produced a second batch And again, 100% Headspace moisture analysis was done on the optimized batch that was produced with the optimized cycle, and you see a much more homogeneous batch. Interestingly enough, you do see outlying samples that did not dry for some reason. But the point that I wanted to make here is that the total analysis time for both these batches was less than a day. And for those of you who use titration measurements, you know that this would take a lot longer to do using titration, and you would also lose all of your product samples because the titration is destructive. So for a relatively little amount of work, you can get a tremendous amount of insight into the process and the final product quality here, the final moisture levels. Another application for using water vapor measurements would be to characterize your freeze dryer. In this particular case, this plot shows a moisture map of a shelf of a pilot freeze dryer. So here you're tracking the vial location on the shelf when you take them out, and then performing 100% Headspace moisture analysis on the samples to produce a shelf moisture map. And here you see the well-known edge effects here, leaving a wet spot in the center of the shelf. And you see in the upper right-hand side, this client also did a correlation with the water vapor levels that they were measuring. And on this particular shelf, they were seeing samples with values of 1 to 4% moisture by weight. Here is a moisture plot from the same pilot freeze dryer, the same shelf, using exactly the same cycle, but now the heat transfer to the vials was improved simply by removing the trays. So now the vials are directly on the lyo shelf, and you see that by improving this heat transfer using the same cycle, you get a much more homogeneous batch. You also get a much tighter range of moisture levels. So in the upper right-hand corner, you see now that samples were produced on this shelf between 1 and 1.8% water by weight. And again, the effort needed to do this was very little. So the total analysis time per shelf of this pilot freeze dryer is less than an hour, and you can see exactly how this freeze dryer is performing and what kind of moisture values the finished product has. Okay, just a few words about moisture content and water activity. So here is a vial, we'll call it product A, and you see that most of the water, most of the moisture is bound in the lyo cake, and you have a little bit of free water in the Headspace. Now, you can imagine another formulation, and this one has equal ratio of bound and unbound water, product B, but you could also have a formulation which has mostly free water in the Headspace, or let's say a higher ratio of free water in the Headspace. All of these vials actually have the same total moisture content, but you can see that water activity increases to the right. And actually, it's the free water that is able to interact with the API and cause chemical reactions or chemical degradation. So again, the total water content is equal in all these product vials, but the ratio of bound to unbound water is different. So hopefully this picture gives you an idea of comparing moisture content to water activity. I'd like to hand this over to Rita right now. We're going to do a little interactive poll, so please take a little time to answer these questions, and we'll integrate the answers into the rest of the presentation. Over to you, Rita.
Thanks, Derek. As he noted, we actually have 3 polls we're going to ask our audience members, and to make your selection, just click directly on the screen and click Submit. So here's the first one. Are you working in or most closely connected to development, manufacturing, quality control, or regulatory? Make your selection and click Submit. Again, the question is, are you working in or most closely connected to development, manufacturing, quality control, or regulatory. And we'll give you a few more seconds to make your selection. Okay, now let's look at the results. All right, so it looks like almost 60% of the audience is involved with development, and the rest sort of involved in manufacturing and quality control. Only a few regulatory responses. So, Derek, any comments on that?
Yeah, that's I think what I expected. And again, in the second half of this presentation, I'll show some industry case studies, which hopefully will apply to everybody here. So it will cover all parts of the product life cycle.
Okay, thanks. Let's move on to our second question. And again, make your selection directly on the screen and click Submit. In this case, you can select more than one answer. So, which moisture methods do you or your site use for lyo products? Karl Fischer titration methods, thermogravimetric methods, Headspace moisture analysis, or water activity determination? Again, you can select more than one answer. Just click directly on the screen. Which moisture methods do you or your site use for lyo products? Karl Fischer titration methods, thermogravimetric methods, Headspace moisture analysis, or water activity determination? So again, give you a few more seconds. And now let's look at these results. All right, so it looks like Karl Fischer is far and away the most popular method, with some interest in some of the other areas. Derek, your feedback on this?
Yeah, so again, as expected, and we talked about this in the introduction, 30 years ago, most people used the gravimetric methods. We've seen the industry evolve to Karl Fischer titration. So yeah, again, most moisture determinations are being done with titration. And then you see Headspace moisture analysis and water activity coming up there. Some people are trying to find better ways to do moisture analysis. So yeah, I think that's what I expected.
Okay, thank you. Let's go move on to poll question 3. Okay, and again, make your selection. Only one choice this time, and click Submit. So the question is, what factor is most important when considering an improved moisture method? More accurate results, non-destructive analysis to save the analyzed product, higher throughput, or the ability to analyze statistical sample sets with more robust moisture data? So again, the question is, what factor is most important when considering an improved moisture method? More accurate results, non-destructive analysis to save analyzed product, higher throughput, or the ability to analyze statistical sample sets with more robust moisture data. So again, make your selection and click Submit. And now let's look at these results. So we do have some variety here. About half are looking for more accurate results, but non-destructive analysis is also important. So Derek, again, your thoughts?
Yeah, that's very interesting. Titration, as I mentioned, does have some challenges with possible moisture contamination, and then the question is, what is that number telling you? So people want some more accurate results. It's interesting that I think the industry also is used to working with small sample sets because of the methods that are available. So I think the higher throughput and the ability to analyze statistical sample sets, I had expected that also to be a little bit low, but we see that increasing in the industry. So, okay. Now, thank you very much for these polling questions and your answers.
Thanks, Derek, and the presentation's back in your hands.
Okay, so let's move on here. Again, coming back to moisture content versus water activity. This is, as the poll showed, most people are used to talking about moisture content, and the concept of water activity actually is not new, but it's not used as much, certainly in the area of sterile pharma or sterile dry product. And so I just wanted to spend a couple slides on water activity. So again, as we talked about, moisture content is how much water there is in a given material. Water activity is if you look at the formal definition, is the partial vapor pressure of water in a substance divided by the standard state partial vapor pressure of water. And the water activity indicates how much free water is available for chemical reaction. An example, a water activity of 0.8 means that the vapor pressure of that material or that substance is 80% of that of pure water. And here's a quote from a website here, wateractivity.org, and it says, because water is present in varying degrees of free and bound states, analytical methods that attempt to measure total moisture in a sample don't always agree. Therefore, water activity tells the real story. So, a little bit of a provocative statement, perhaps, but I think this concept is becoming more and more interesting for the pharma industry, and that's shown by the introduction of a new USP chapter on water activity, USP 922, and this is scheduled to be implemented in the first quarter of next year. So a draft version was available a year and a half ago. It went through a comment process, public comment process. A second draft was made available, and I believe they are working on this second draft now to finalize it at the beginning of next year. And again, there's a similar quote to what we just read from this USP chapter. It's a little bit much, a little bit of text, but I do want to read it for you. And it says here, total water content is an important quality attribute. However, water may be allocated in more than one compartment within these materials. Some of it may be tightly bound and not available to participate in chemical, biochemical, or physicochemical reactions, for example, hydrate salts, whereas some of the water may be more loosely bound and more freely available to participate in reactions. It is important to establish what fraction of the total water is contained in the latter category, and the determination of water activity provides this information. So this is a USP chapter which is encouraging the use of a water activity measurement, and I think this is being picked up a little bit more in the oral solid dosage area, but it has a list of applications for water activity, and I'll just read a few here, I think, which apply in general to dry product, and certainly freeze-dried product is included here. So the determination of water activity aids in decisions during ingredient and product processes design, ingredient selection, packaging selection, and product storage. These include reducing the degradation of active ingredients within product formulations, especially those susceptible to chemical hydrolysis, providing a complementary method to the Karl Fischer titration for monitoring changes in water content, controlling and monitoring physical, chemical, and microbial product stability. Again, if you have not seen this chapter or are aware of this upcoming chapter, the draft version is available, and you can read through this prior to the implementation and beginning of next year. So I wanted to start with an industry case study, which is actually a water activity method study. In this particular study, a manufacturer was looking to replace Karl Fischer titration with a rapid non-destructive method for lyo product moisture determination. And they chose to do some studies using Headspace moisture analysis, and they were looking to assess the correlation between the water vapor levels in the freeze-dried vial and the degradation of a small molecule API. So how is this study set up? Some sample sets with a range of cake moisture were prepared, ranging from 0.5% to 3% water by weight. The initial moisture in the headspace of these samples were measured and at a fixed temperature that water vapor pressure is the water activity of that product. And since the measurement is non-destructive, these same samples could then be put on stability. So the samples on stability were monitored for degradation of the API, measured with HPLC, and then a correlation between water activity and API degradation was determined. So here's a little diagram of the study. Again, you see the 5 moisture groups there, from 0.5% to 3% water by weight. And then you see that these samples were stored on stability at 3 different temperatures, 25 degrees, 40, and 60. And you see down below that for these targeted moisture groups, you see what the corresponding water vapor levels were in these sample sets, and those range from 4 up to 14 torr. So here are some of the stability results, and what we're plotting here is the degradation of the active component. In this case, it's the 60-degree data over time. And you can see, as expected, the high moisture samples at 3% water by weight were almost fully degraded or suffered from full degradation at about 4 weeks. The low moisture samples at 0.5% water by weight after 4 weeks and being stored at 60 degrees showed a slight degradation. Now, you can take these stability results and then plot them versus the water vapor content in these samples. And this is the correlation that you get plotting the degradation, or the degradation constant, the log of the degradation constant, so the rate of degradation versus the water vapor pressure. And this correlation is quite amazing. So you see an R squared of 0.99. You can clearly see the 5 different moisture groups just lying right on that line. So those were the moisture groups from 0.5% water by weight up to 3% water by weight. So what this data actually tells you is that a water vapor measurement, or a water activity measurement, exactly predicts the stability of this particular formulation. So there's a potential here to replace the titration measurement with a rapid, non-destructive technique for lyo moisture determination. So this water vapor measurement will allow you to exactly determine the product stability, and also from a pragmatic point of view, being rapid and non-destructive has a lot of advantages. You can see here the full stability results also at 25 degrees and 40 degrees, and you can see that the correlation of water activity to product degradation is very good for all temperatures and moisture content. We're almost ready to submit a paper on this study, which I hope will then be available soon. So just a couple more examples of using water activity to determine product stability. Here you see a presentation, or the title slide of a presentation given by GSK Vaccines a couple years ago, where water activity was used to determine the stability of freeze-dried vaccines, so live viral vaccines. The title was How to Use Water Activity Measurements and Mathematical Modeling to Evaluate the Stability of Freeze-Dried Vaccines. And I just wanted to point out the mathematical modeling in this title, because water activity is actually a thermodynamic property of a material. And so you can start with fundamental equations and use water activity as an input in those equations and work out and model the stability of a dry product. And so very useful, again, with predictive stability modeling, which I think is an area that's a very intense area in the oral solid dosage form. But you see here GSK also using this to predict the stability of freeze-dried vaccines. I've mentioned oral solid dosage forms here. People there have used the water activity measurement for some time now. And here's an example of that, also with a reference to an article there if you would like to get more detail. So water activity measurements at least we know of a number of companies that have used or implemented the water activity measurement for solid dosage form in their QC release, but also their stability programs. So these previous slides introduced Headspace moisture and the idea of water activity and demonstrated that a formal water activity method could be developed, which actually predicts the stability of dried product, or in particular a freeze-dried product. I had a few examples at the beginning of the presentation which showed how the water vapor measurement can be used to gain insight into the process or the freeze dryer. And so now I want to have a couple industry case studies that show how the water vapor measurement is a useful analytical tool in supporting scale-up activities and the optimization of commercial batch production. So here is some data, and again, you will recognize it as a moisture distribution plot of a freeze dryer shelf. And in this particular case, the client wanted to do two things. They wanted to test a cycle on two different freeze dryers. They weren't sure if it was an optimum cycle or not, so they wanted to see how well the cycle performed, but they also wanted to show or check if the two freeze dryers were equivalent when this cycle was used. So here is a moisture plot of a shelf from both freeze dryers. These were both pilot scale, but I believe at least one of them could go up to batch sizes of almost 10,000 vials. And from this particular moisture plot, you can see, you can make a couple of conclusions here. Number one, the cycle is not optimum because you see some inhomogeneity here, and two, the freeze dryers are certainly not equivalent. If you look at freeze dryer 1 a little bit more closely, so the formulation model was a 5% sucrose solution in a 6-hour vial with a 2.5 ml fill. Full Headspace moisture analysis of a shelf gave water vapor levels that correlated to a range of 0.8 to 1.4% moisture by weight, which is not a bad range, but it is inhomogeneous in the sense that the edge effects are clear in this plot. So you see that on the sides and the front, the samples clearly dried better or faster than the samples in the center of the shelf. And if you compare that to the samples coming out of freeze dryer 2, again, exactly the same solution, the same lyo cycle. And here you see that samples coming off of this shelf measured water vapor levels that correlated to 0.6, a range of 0.6 to 2.2 percent moisture by weight. So a more inhomogeneous batch, larger variance, and a larger range of moistures. The cake appearance was acceptable with some minor shrinkage in some vials. And here you see clear edge effects front, back, and side, where again, those samples are drying faster than the samples in the middle. So this client ended up doing two things to try and optimize the situation. Number one, they optimized the cycle, so they chose certain parameters to change in the actual cycle. And they also added 2.5% BSA into the formulation to simulate the freeze drying of a biopharmaceutical. And then they ran the optimized cycle again on both freeze dryers, freeze dryer 1 and freeze dryer 2. Again, did a moisture mapping on the shelves. And here you see the results. Clearly a lot better than the first attempt. And so you see that not only are these two freeze dryers producing very homogeneous batches now, but they are also equivalent to each other. And so here you can see, I think, a good example of how Headspace moisture analysis of statistical sample sets enabled, number one, an efficient cycle optimization, and number two, a clear demonstration of freeze dryer equivalence. And again, if you think about using the traditional methods to do this, Obviously, very challenging if you have to analyze this number of samples either with titration or gravimetric methods, and obviously then you would also lose all the samples that you analyze. Okay, another industry case study here, and you can see here that this case study was actually presented as part of a presentation at the PDA Pharmaceutical Freeze Drying Technology Conference a couple years ago in Sevilla, Spain. This particular case study again comes from GSK vaccines, and the situation here was an old freeze dryer needed to be replaced in an existing facility during running operations. The site wanted to double the capacity, so scale up from 14 square meters to 28 square meters. And during this replacement of the old freeze dryer, they wanted to introduce some improvements, such as points of fill filtration, also get a very good determination of intra and inter-batch variability of the new freeze dryer, and also take a very systematic quality by design approach to comply with recent FDA guidelines. So they chose Headspace moisture determination as an analytical tool to perform a design of experiments in small scale. And so here what they did is working with some pilot scale batches, they performed 100% moisture analysis to determine intra-batch variability. These moisture results were also used to analyze the impact and interaction of critical process parameters with inherent product variability. So again, having statistical data allowed a very good statistical analysis for this. The results also enabled them to determine a suitable placebo for the full-scale validation runs of the new freeze dryer. And in addition, all of these samples that were used for moisture analysis in this design of experiments were still available for other destructive tests, which allowed them to do one-to-one correlation of moisture to other parameters. And of course, this would not have been possible if the samples had been analyzed again with titration. So here are some results from this case study. Here you see that they use different lyo cycles. So what we're plotting here is the Headspace water vapor levels of various sample sets that were produced with different cycles, but also different formulation or different placebo configurations. And so this allowed them to verify the design space of the process. It also showed that you see some comparable moisture patterns between the different placebo configurations. And with this statistical data, they were able to do a DOE analysis and very clearly show which product-specific attributes showed a high dependency on the moisture. And these results were then used to define a suitable placebo for the full-scale production runs or validation runs of the new freeze dryer. So conclusions from this case study, they demonstrated that a proof of concept for the full-scale runs was possible using this DOE on a pilot scale approach, and this allowed them to minimize the project risk and also the number of full-scale runs that they needed for the full-scale validation. It enabled them to determine a suitable placebo, and this mitigated the lack of drug substance and also minimized the cost because they did not have to use real drug substance for a lot of their development activities and validation activities. It verified a robust design space, so they have this data available to them, which they can use to argue and to show and demonstrate that they have a robust design space. And it allowed them to build up a lot of process knowledge about the interaction of critical parameters and their effect on the intra and inter-batch variability. Okay, so that was a case study of a pilot scale approach moving towards validation of the commercial process. I just wanted to spend the last case study here on actually looking at moisture distributions of a real commercial batch, and in particular to show how you could possibly use this to save lyo capacity at a freeze-drying facility. So here, this data graph is showing the moisture distribution of a commercial batch of product. The batch size was almost 14,000 vials. They were 100% inspected for headspace moisture levels. And what you see here is that is a fairly conservative cycle. So almost all of the product was very dry. So you see very low headspace moisture values here, except for 2 vials, which did not dry very well and in fact stayed very wet. So you see those out there on the right-hand side of the graph. And this is the inefficiency of the freeze-drying cycle, in that you have vials that are drying at different rates, and so you have to keep the process running until the last vial is dry enough. And to do that, most people will over dry or design cycles that are quite conservative. And you see in this case that it pretty much worked except for these 2 vials. So you could say that waiting for the last vial to dry didn't work. Now, if you look more carefully at these results, for this particular formulation, a typical specification was 3% water by weight, and that, again, in this particular case, corresponded to roughly 3 torr of headspace water vapor. So you see that most of the product is very, very far away from the specification. In fact, fairly dry compared to the specification, and only those 2 very wet vials obviously are not do not meet specification and are very wet. But here you can see that a lot of extra lyo capacity was used to dry these vials. And in fact, you could see batch monitoring as a way to save lyo capacity. If you can shorten, for example, your secondary drying time, and if you just monitor your batch moisture distributions and make sure that they are still below spec, you can imagine that you could win back significant lyo production capacity if you have the capability to produce this kind of data. And in this particular case, the data was collected in a fairly straightforward batch headspace inspection process using one of those automated machines that you saw a picture of at the beginning of the presentation. And it only took an hour and a half to generate this data with a headspace inspection machine running at 150 vials per minute. So I think some very interesting things that you can do if you are able to monitor the moisture distribution of a full batch of product. Okay, so I hope that these case studies have made a good argument for a water vapor measurement being a useful analytical tool for process development and scale-up, and for producing statistical robust moisture data. Why would you want to generate this kind of data? Well, current things that are driving quality in the pharma industry, regulators are asking for more analytical data. They want decisions that are data-driven. The quality by design approach asks for a lot of data in the early parts of the product life cycle so that you can design a robust commercial process. There's always discussions about how robust validation data is. The concept of monitoring and controlling in manufacturing is also a very hot topic. If you look at current guidance, such as the FDA process validation guidance, you will read things like having statistical confidence in the process, so generating data that allows you or that enables statistical confidence in the process. Or here's another quote out of process validation guidance: the collection and evaluation of data from the process design phase throughout production, which establishes scientific evidence that a process is capable of consistently delivering quality products. And this is very difficult, of course, in the freeze-drying process because of the nature of the process. And I think there's a lot of very good work going on to use control methods of the actual lyo cycle or the lyo process itself as the freeze dryer is running. But I think there is a little bit of an underappreciation of what you can do with the finished product analysis. So if you are able to do moisture analysis on statistical sample sets, you can also get a lot of insight into the process and optimize by looking at that robust moisture data. So there's the bullet point analysis of statistically relevant sample sets, enables decisions that are justified by science-based data. And to do that, really, you have to increase your analytical toolbox. So we talked a lot about the standard methods being titration, gravimetric. If you can add methods to your toolbox which enable more robust data generation, then you can, I think, enable much more efficient and robust characterization and optimization of the process and also your freeze drying equipment. So in summary, we've made this point that the current moisture determination methods lack the ability to really do statistical sampling on the moisture of dry product. Headspace moisture analysis is a potential analytical tool that can be used in all phases of the product life cycle to determine the moisture content and also the product stability, if you correlate to product stability of dry product. And it's been demonstrated that Headspace moisture analysis can also be used to actually develop water activity methods, and water activity as a measurement is gaining traction in the industry, and I think more and more people will be looking at this in the future. Thank you very much for your attention. It was a lot of data. I hope, and I apologize if I went through it too quickly. If you do have some questions, there's more information at our website. There are also places where you can request more information or leave questions behind. I think right now, actually, we do have some time left over for a Q&A session. So I will hand this back over to Rita.
Well, thank you very much, Derek. We will get to a few questions, but a reminder to our audience, you can submit your question by typing it in the Q&A box, which is located on the right-hand side of the presentation window. So here's the first one, Derek. So what is the difference between NIR and laser-based headspace analysis for moisture determination?
Yes, that's a good question, and I mentioned NIR at the beginning, and I realize it probably would have been helpful to include NIR as a moisture technique in that polling question. We could have seen how many people are using NIR at the moment. So and one of the reasons I didn't include NIR is essentially NIR is linked back to titration. So when you use an NIR measurement, you are collecting spectra, so you don't get a number. And so you have to calibrate that spectra in some way. And people will generally calibrate the spectra with titration. So NIR is not a primary measurement, it's a secondary measurement. So that's one difference between the two measurements. A water vapor measurement gives you a number, and it's a primary measurement. You don't have to correlate it to anything if you don't want to. And these calibrations of NIR can be tricky. To get a real robust, stable calibration to use NIR is some amount of work, and you also have to monitor to make sure that that calibration remains stable. If you change, for example, your vial supplier and it's still the same clear tubing 2R vial, just changing vial supplier will sometimes throw off the calibration of your NIR technique. So those are some of the, I think, some of the reasons why NIR hasn't quite been able to displace the standard methods.
Okay, thanks. Here's the next question. Are there limitations on the sample container size that can be analyzed by Headspace analysis? So, for example, can product lyophilized in a 1 liter bottle be analyzed?
A 1 liter bottle is quite large. We do have systems that are that are analyzing 1 liter bottles, but we had to make it a custom system. So the answer is theoretically yes, you can measure all sizes of containers. The standard sizes that we measure in our standard instruments range from syringes, half ml syringes, up to let's say 200 ml bottles, and then we have some custom configurations for larger containers.
Okay, thanks. Here's another question. So what's the difference between Lighthouse moisture, pressure, and water activity instruments?
Okay, so there's somebody who knows a little bit about our instruments, because we do indeed have 2 platforms that allow you to measure Headspace moisture, and one of them is indeed, we call it a Headspace moisture pressure instrument, because it will measure the water vapor levels and the vacuum levels in a vial. And sometimes that's useful for freeze-dried product, because you will have freeze-dried product at an under pressure, and measuring both those parameters could be useful. The main difference between the 2 instruments, though, is that the water activity analyzer has a temperature-controlled sample chamber, because a water vapor measurement is not the water activity of the material unless you measure at a defined temperature. So water activity is always defined for a specific temperature. So our water activity analyzer actually has a temperature-controlled sample chamber, which allows you to set the temperature and make sure that you take the measurement at a specified or defined temperature, and it then returns a water activity value. Of course, with the Headspace moisture system, if you're just making room temperature measurements, you could, in principle, say that Headspace moisture value is the water activity at whatever the room temperature is.
Okay, thank you. Is there an impact of water content of the stopper over time?
Yes, so that's a very good question. And in fact, a lot of the stopper suppliers have used these platforms to try and understand how much moisture has been retained in the stopper after it's been treated, and how much of that moisture actually escapes into the vial. And this will depend on, well, the drying process, obviously, and will also depend on the exact rubber formulation. So in principle, you will get a slow outgassing of water vapor from the stopper over time, but it's a very slow process. But for moisture sensitive product, it could be something that you need to take into account.
Okay, thanks. Here's another question. What is the ideal height of cake and headspace in a vial?
Ooh, so I guess I would ask ideal for what? If it's about being able to make a good measurement, to make a good headspace measurement, we simply need, let's say, a minimum of 3 to 5 millimeters of headspace in the container. And in general, when you're freeze-drying product, the liquid fill might be a little bit high, but freeze-drying, obviously, you end up with a freeze-dried cake, which is lower in height than the original fill. So normally, almost all freeze-dried product is very straightforward to measure with headspace analysis.
Okay, thanks. Next question. Does colored glass impact the measurement?
Yes, a very good question. So colored glass, you're talking about amber glass for light-sensitive products. You could also ask the same question about molded glass. If you have molded glass, very bad glass quality, does that affect the measurement? And the answer to both of those is no, it doesn't affect the measurement. Or to be more precise, you can make a measurement in these containers, but the color and the molded characteristic could cut down on the transmission of light through the container. So this might mean that your signal to noise ratio is not as high as clear tubing glass, which means that the standard deviation of the measurement could be a little bit higher. So you might get higher standard deviations in the measurement, but it's possible to do this analysis on both colored glass and molded glass.
All right, we have time for one last question. If our analytical method is Karl Fischer and we want to switch to laser analysis of the Headspace, how can we validate this method so that it is equivalent to the method by Karl Fischer?
Yeah, so I think there are basically two main approaches that you can use. For an existing product, I think when you replace an existing method, you want to show that the new method is as good or better as the old method. So then you would set up some correlation studies. You could do some correlation experiments that similar to the ones that are shown, correlating the water vapor level to the moisture content value as measured by Karl Fischer. And if you get a very good correlation, you can compare those two measurements and also produce data to show that your headspace measurement is performing as well or better than the existing measurement. If you're working with a new formulation, you do have the possibility to correlate the headspace moisture directly to the product stability. So there, you don't even use titration. You simply set up a study and try and show that the water vapor levels that you measure in that product allow you to predict the product stability. And then you define your specification actually in terms of water vapor or water activity if you want to express it as water activity. And that was exactly what one of these case studies showed, is that you don't even need titration if you can show that the water vapor actually correlates directly to product stability.
All right, well, thank you very much, Derek. Unfortunately, we're out of time. If we didn't get to our audience questions, it'll be answered offline. I want to thank the audience for attending and participating in today's event. And I'd like to thank our sponsor, Lighthouse Instruments, for making today's webcast possible. You'll receive an email alerting you when this webcast will be available for replay. And I invite you to forward that announcement to your colleagues who may have missed today's live event. So thank you again for joining us, and have a great day.